This video is for educational and informational purposes only. The content is based on personal experiences and is not medical advice. Always consult a qualified healthcare professional for personalized medical guidance.
Standard of care for glioblastoma is responsible for the overall poor survival and death of these patients. Absolutely. I'll stand by that because I have the scientific evidence to support that. They're all the same disease. Brain cancer is not this different from breast, colon. They all have the same problem with mitochondria. They're all dependent on glucose and glutamine. It's not 100 diseases. It's one singular disease of energy, of dysregulated energy production. Now, how complicated is that? The future is this. What I just told you is the future. And we're going to, maybe not me, but others will perfect it to the point where cancer will not be considered a fearful disease. But we have people will start getting healthy when you're killing their cancer cells, not making them worse. Hair fallout. What are you talking about? The current cancer paradigm is based on cancer being a genetic disease. And if you go to the National Cancer Institute website, it says cancer is a genetic disease comprising over 100 different types of disorders, all driven by different kinds of genetic mutations. The new data, if you look carefully at the data, it does not support this information from the National Cancer Institute. As a matter of fact, to accept cancer being a genetic disease, one would have to be either cognitively challenged or a hopeless ideologue, because the evidence clearly shows that we have some cancers that have no mutations. We have driver mutations in normal tissues. Driver mutations were thought to be those that really led to the dysregulated cell growth, like P53 and some of these others. And now we find these P53 mutations in normal tissues of people that never have dysregulated growth. And the nuclear transfer experiments, which have been repeated in many different types of cancers under different experimental paradigms, where the nucleus of the tumor cell is put into a normal cytoplasm, and you don't get dysregulated growth, where if the normal nucleus is put into a tumor cytoplasm, you get dysregulated growth. This information overwhelmingly says cancer cannot be a genetic disease. And what Warburg had first done and what I've pushed over what he had done shows that cancer is a mitochondrial metabolic disease, not a genetic disease. And this is the reason why we're not making any progress. You have to realize there's two things here. Number one, we've put billions of dollars into the Cancer Genome Atlas and thinking cancer is a genetic disease, and it hasn't had any major impact on reducing deaths. Consequently, because that has not come to fruition, we continue to poison and irradiate people, because the idea was that if cancer were a genetic disease and we were able to target the genetic mutations, we could get rid of the poisons and radiation. But because cancer is not a genetic disease, we can now have to continue with poisons and radiation and all the other things that are being done, like immunotherapies are based on cancer being a genetic disease. Some people do well on immunotherapies, but you can just as well die from the therapy as being cured from it. And a lot of people don't even respond to it. So clearly it's not moving the needle. And you're right, each year we get more and more dead people, and we're not making any progress. I don't care what anybody says. 1,700 people a day dying in the United States, 70 an hour, and continues to increase year after year. It means that we have made zero progress. So when you drop the death rate by 30%, then you know you're starting on the right path. But we're not doing that. So, I mean, people can I look at it as a monumental failure. And it's a failure because the theory of what the disease is incorrect. And when you have an incorrect theory, you're not going to move the needle. Well, it comes from a number of different places. It came from the fact that in the 1950s, when Watson and Crick were awarded the Nobel Prize for the structure of DNA, and people had always, it actually starts with Theodore Boveri in 1914, when he speculated that there was some abnormalities in the nucleus of cancer cells based on studies in sea urchins. which doesn't make any sense to me. And one of his friends, I think his name was Hammerschlag, told Bovary, Theodore Bovary, that this is all nonsense. As a matter of fact, Bovary wrote a big article on this and was very apologetic about cancer being a genetic disease. He said, I have no clue about what cancer is. I don't know anything about it. Everything is pure speculation, and I could be completely wrong. Yet he was anointed as the father of the somatic mutation theory. So, but he himself said, I don't know anything about cancer. And then others came along and started to cancer damage to the nuclear genome. It was always seen in not always, but recognized, often recognized in cancer cells. But it wasn't until the early 50s when Watson and Crick found the structure of DNA, and then you found structural abnormalities in the nucleus of cancer cells, structural DNA abnormalities, and everybody was, gene think, genes control everything, and you have mutations in genes, and that's going to lead to dysregulated cell growth. And the more and more scientists jumped onto this, these oncogenes and tumor suppressor genes and all these kinds of things, but failing to recognize that the gene mutations themselves are arising from the damage to the oxidative phosphorylation. So the genes are not the cause, they're the effects. And once you realize that, then it's such an embarrassment and tragedy to realize that the resistance to change and the attachment to this incorrect theory has led to all these poor folks being killed by not only the disease, but the treatments. I don't know. I think half the people that are treated for cancer die from the treatments. I mean, it's horrific. You don't poison and irradiate people to make them healthy, right? This is what they do. Surgically mutilate them, remove breasts and colons and lungs. I mean, this is the way we're treating cancer patients. As I said in my other interview, it's medieval. Nothing could be more medieval than the current cancer paradigm of treating people. When the oxidative phosphorylation, the ability of the cell to generate energy through oxygen, when that becomes compromised, these cancer cells produce reactive oxygen species, ROS, and they are carcinogenic and mutagenic. So the mutations and damage we see in the nucleus come from, in large part, the ROS. So there's a clear link between damaged respiration and the mutation. So we've been chasing tail. We've been chasing downstream effects. And when you chase downstream effects, the outcome is 1,700 people a day dying, and it's not getting any better. Well, we know that because mitochondria control the differentiated state of the cell. Mitochondria can determine, they control the cell cycle through calcium signaling changes that our mitochondria does this based on the proton motive gradient in the mitochondria. So when cells need to divide, they don't divide out of control. Mitochondria maintains division under control or maintains the quiescent state of the cell. When that organelle loses control over the energy metabolism of the cell falls back on fermentation metabolism, which is energy without oxygen. This is the kind of energy that existed on the Earth before oxygen came into the atmosphere 2.5 billion years ago. All the cells at that time were dysregulated in their growth and driven by a fermentation metabolism. So the cancer cell is doing nothing more than falling back on these ancient pathways leading to a dysregulated cell growth, because the mitochondria that's supposed to control this process loses control of this due to a number of different insults that could damage the efficiency of oxidative phosphorylation. So there's a two-step process. One is a gradual, slow interruption with oxidative phosphorylation, maybe from intermittent hypoxia, systemic inflammation, some viruses, some chemical carcinogens. Any one of these provocative agents can lead to a gradual disruption of oxidative phosphorylation, leading to a gradual increase in what we call substrate level phosphorylation, or energy energy production without oxygen. So substrate level phosphorylation can produce energy without oxygen. So the cell and the default state of mammalian cells is proliferation, and 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 the control of proliferation is directed by the mitochondria. So if the mitochondria gradually become dysfunctional energetically and the cell falls back on its energy default state, which is proliferation, then the cell is in a dysregulated state. So the mitochondria control the destiny, the differentiated state of the cell. And then you get all the secondary mutations, a lot of other secondary. They control the apoptotic signaling. What does that mean? Programmed cell death. So when our cells are injured, the mitochondria release cytochrome C, leading to a cascade and program cell death. In other words, the cell is programmed to die because it has damage or something along these lines. Mitochondria control that. They control the kill switch of the cell. And when the mitochondria become defective, the kill switch is broken. So the cell bypasses the normal programmed cell death mechanism, continues to proliferate out of control, driven by a fermentation metabolism. If people understand evolutionary biology and general biology, and what I'm saying makes perfect sense because it's all supported by scientific literature. But when you are indoctrinated to think cancer is a genetic disease, you don't think about any of the stuff that I just spoke about. And therefore, you have this perpetuation of dead people. So until this, what I just said, is understood and recognized more clearly, then we're not going to make any changes. Yeah, and that's one of the things in the hallmarks of cancer. It's the failure to recognize apoptosis. So and different and dysregulated cell growth that comes from the fact that the cells are fermenting. So all of these things, you can go through all the hallmarks of cancer and link all of the hallmarks clearly back to mitochondrial dysfunction. I did a deep dive on this whole thing, spent a lot of time. You have to go back in the scientific literature and look at electron microscopy because the electron microscope wasn't perfected until the 1950s, 60s. People, started looking at the ultrastructure of cells because on light microscopy you really can't see much. Mitochondria are thread-like organelles that exist in the cytoplasm, but in order to really look at them carefully and in detail, you need electron microscopy. So when you go back and look at a lot of these guys that were looking at the ultrastructure of tumor cells were just pointing out what they saw. There was a lot of cellular abnormalities, but a lot of the mitochondria were abnormal in structure. They had sometimes no cristae, the inner membrane elaboration of the organelle. Sometimes there were few mitochondria. Sometimes the cytoplasm was packed with abnormal looking mitochondria. There was all kinds of morphological abnormalities. And then, of course, when cell biology started to isolate mitochondria in pure form from tumor cells, it was clear that there was all kinds of functional abnormalities in these organelles. And we here at Boston College, we studied cardiolipin, which is a very specialized kind of lipid in the inner membrane of the mitochondria that plays a very significant role in the efficiency of the electron transport chain, which is ultimately the efficiency of oxidative phosphorylation. And we found massive changes in number and structure of the cardiolipin, meaning that this lipid, which controls the efficiency of mitochondria, was always abnormal. In fact, no one has found a tumor cell yet with a normal structure and function of either mitochondria or cardiolipin. So this clearly tells us that these mitochondria are not functionally effective. They're insufficient in their ability to produce energy through respiration. Therefore, the cell falls back on a fermentation mechanism, which is energy without oxygen and a dysregulated growth. It's not that complicated if you understand the biology of the situation. Yeah, sometimes you do. Sometimes it becomes excessively steep, and other times it's dissipated. There's uncoupling proteins. The oxygen that enters the mitochondria, what we and others have found, is not linked to ATP synthesis. So our big discovery that Warburg did not know about, nor does most of the people in the cancer field, They don't know that there's two ways for a cell to get energy through substrate level phosphorylation. One is in the glycolytic pathway, the Embden-Meyerhoff-Parnes pathway, which is the cytoplasmic pathway that produces energy and the payoff phase of glycolysis. And many cancer cells dump lactic acid into the microenvironment, which is the where pyruvate, instead of going into the mitochondria and being respired, is reduced to lactate, which then accumulates in the microenvironment, leading to acidification. Tumor acidification is common in many kinds of cancers. Warburg knew only about that. Our discovery was that there is a substrate level phosphorylation process inside the mitochondria itself. So this is an organelle that can both respire and ferment under different kinds of conditions. And no one knew about that. That's our big discovery. Paper just came out recently, late December 2024, where we're getting tremendous recognition about on this study. For the first time, people are realizing that these mitochondria can ferment glutamine. That glutamine amino acid can be metabolized through the mitochondria to succinic acid, which also contributes to extracellular acidification. So now, here, why everybody said Warburg was wrong? Why so many people thought Warburg was wrong? Because Warburg himself thought that oxygen consumption in the cancer mitochondria was also linked to oxidative phosphorylation. And his critics said, well, how can Warburg be right saying that oxidative phosphorylation is the origin of cancer when cancers are taking in oxygen sometimes as high as normal tissue? And this created a tremendous amount of controversy because yes, the cancer mitochondria does take in oxygen, but it's not contributing significantly to oxidative phosphorylation. They are producing ATP through the mitochondria, but through a fermentation mechanism. This is the big new thing. The new thing is the cancer mitochondria can ferment and they dump out succinic acid. So now we put the parts together from Warburg. Warburg was right. Cancer is a mitochondrial metabolic disease. He just didn't have all of the details together like we have today. Now it becomes irrefutable. Cancer is a mitochondrial metabolic disease driven by fermentation metabolism because oxidative phosphorylation is inefficient. So we know this, and therefore the cell proliferates out of control because the organelle that's supposed to control this is not working. And now with the knowledge that this is a fermentation problem, and that the two fuels driving the dysregulated growth of the sugar glucose and the amino acid glutamine, and we know because that mitochondria is defective, they can't use fatty acids or ketones. Now we have a solution to the problem, right? Transition the whole body over to ketones. The tumor cells can't use them. They store them in the cytoplasm. And then you target simultaneously together the two fermentable fuels. And these cancer cells are checkmated. So they can't use anything else. And how do we know that? We tested all this stuff. We interrogated these cells and to see what they can use and can't use. They can only use glucose and glutamine. Yes, they can use a little bit of other things, but the term is logistics. In order to replace the high energy efficiency of oxidative phosphorylation, you better make sure that the alternative fuels are present in the microenvironment in large quantities to run the dysregulated growth. And some of these other amino acids are not there, only glutamine. So clearly, we know what's going on. It's just that, unfortunately, the world doesn't know it or refuses to accept it. And therefore, we got 1,700 people a day. It's tragedy because we can stop it. We can significantly reduce it with what I told you. But the clinical trials, we have to have people that are knowledgeable. So we have to train people to understand what I just said, understand how and we just put this big paper in, a big protocol paper for glioblastoma that goes through a lot of what I said by my colleague, Dr. Thomas Duryea, next door here, works with me. And we have 25 other nutritionists, dietitians, scientists, MDs, physicians, all kinds on this paper. Once they realize that this will be the strategy for managing cancer, we can adapt it for all the cancers. They're all the same disease. Brain cancer is not this different from breast, colon. They all have the same problem with mitochondria. They're all dependent on glucose and glutamine. It's not 100 diseases. It's one singular disease of energy, of dysregulated energy production. Now, how complicated is that?